Hello and welcome to a new aquafeed.com webinar, Improving Gut Health through Aquafeeds. My name is Lucia Barreiro and I am the editor of aquafeed.com. Optimal gut health is vital for aquaculture species, and problems can be generated by the feed quality and nutritional status, among other factors. In this webinar, we will discuss risk mitigating strategies using feed ingredients and additives for an optimal gut health, microbiota balance, and performance. Thanks to the support of our sponsor MOTIV, this webinar is open and free. Each speaker will give a 15-minute presentation, and then we will get to a final panel session to answer questions from the audience. To submit a question, please type it in the Q&A box. Special thanks to our assistant editor and webinar manager, Marisa Yanaga, who is making all this webinar works. And finally, it's my pleasure to introduce Hans Boon, who will be moderating this webinar. Hans graduated from Wageningen University in 1987 as biologist, specialized in aquaculture and nutrition. He has been one of the driving forces in the aquafeed division of Provimi, now Biomar, where he held several positions from 1989 until 2008, when he founded his aquafeed consultancy, Aquaculture Experience. Aquaculture Experience is a globally operating advisor of aquafeed and feed ingredients companies. Hans, hand it over to you.
Okay, thanks, Lucia. Thank you all for participating in this webinar. And yeah, well, the floor is basically meant for the 4 presenters today, but it makes me really happy to see a nice bunch of old guys and also some very much youngsters. So there's some veterans here and also some, well, upcoming young professionals in our symposium or webinar today. I have the pleasure of introducing the first speaker, Dr. Nguyen Duy Hoa. I'll call you Dr. Hoa to make it easier and not break my tongue all the time. Dr. Hoa received his bachelor in aquaculture in Nong Lang University in Vietnam in 1994, so also an old-timer already in this business. His MSc in aquaculture in 2003 and PhD in applied biology engineering for shrimp nutrition and domestication in 2009 in Ghent University. He's been working in the industry for aquaculture in Vietnam he has been working for over 17 years in this industry already, 18 years in the National Research Institute of Aquaculture in Vietnam before he joined INVE, Thailand, as key account manager, where he was focusing on health products, so really fitting into our topic today. And he joined Cargill Vietnam, where he's still working in 2013 as a global technical director in 2016. The floor is yours, Hoa.
Yeah, thank you. So can I share the screen now?
Yes, please.
Okay, can you see my presentation?
Yes, we can.
It's okay.
Okay, first of all, thanks a lot Aquafeed for organize this webinar. And also thanks a lot all the participants from different time zoning. So today I want to talk about the topic on the product MOTIV, biobuck active protein for animal gut health and stress resistance. So here's the agenda of my talk. First, I will talk about what is the gut health, and then gut health management strategy, and then why fermentation, and then MOTIV, a bioactive protein ingredient, and then the conclusion. So here's one of the slides I took from Google for human gut health, and you can see the gut health in general is significant impact on the overall health, including the mental health, immune health, and metabolic health. In particular, you can refer to stress, diet, or the immune and stress condition. And also in the Google website, we can have several information on the gut health in human. And at the end of the day, you can see the forming a healthy gut diet is a key and overall for overall health management, not only the gut health. And for gut health management strategy, again, managing diet and stress is a key strategy. And for diets, we talk about the intolerance and the performance, how the animal or human beings accept the food and performance that food into the health or growth performance. And for diets, there's several factors that can impact on the intolerance or in terms of digestion or in terms of in general in performance also. So first is the adaptation, palatability, and digestion also that own impact on the absorption and the digestion also. Safety issue and spoilage is very, very important for the gut health. For example, if you have the food with a lot of mycotoxin or a lot of other toxin like pesticide, for example, that also impacts the gut health. Sometimes I eat the vegetable that contaminated with pesticide. Just some hours later, I got the diarrhea. So very soon, very, very clearly on the safety issue, it's a big impact on the gut health and spoil it, the food spoil it also. When you eat the food that already spoil it, you get diarrhea. So the same animal, the same. And the food with the storage condition also that can keep quality of the feed, quality of the food for the animal to be not spoiled, to be not oxidized, be oxidized to impact on the gut. In terms of nutrients, the anti-nutrient also one of the factors that can impact the gut a lot. It can make the gut inflammation and open the entry for the pathogen. Again, gut health and anti-stress nutrient is one of the factors that can improve the gut. Cooking process also the same. When you cook the rice, if you cook not well, you cannot eat. But if you cook well, you eat well. It's the same for the feed technology. If you cook not well, the feed, you will impact the digestion of the food and other factor, physical factor also. That also leaching the nutrient in the water, and then the animal consume that bad food, and that also impact on the gut health. And not only the diet, the stress can be impacted by the environmental parameter. For example, in the shrimp, if you culture the shrimp out of the pH, out of the low dissolved oxygen or high pH, low pH, and so on, that can also stress the shrimp too much, and that also impact on the gut health and the health in general. Pathogen in the gut also. That also disturb the digestion, disturb and also damage the gut. Diet also. If you feed the animal not right diet, not right nutrition, it also cause the animal stress. So in terms of the managing diet stress, we see that some issue that can impact on the animal gut health or aquatic species also. First is the microbial in the gut. If you have the gut with the not good feed or not good nutrient that favorable for the beneficial bacteria, and then you will have the more pathogen. And how to get that bad gut to the good gut microbiome? You need to feed the shrimp with the shrimp or animal with the prebiotic, probiotic, and you know that fermented protein can provide serve as a prebiotic also. In terms of stress, antioxidant nucleotide, beta-glucan, sorbitol, inositol, some special amino acid like glutamine, leucine, or taurine, those can help to reduce the stress also. Biopeptide is one of the very good for the stress resistant and antibacterial. Vitamin and mineral, of course, is one of the health nutrient, and that can do the very good, the co-factor for enzyme for better digestion. And fermented protein that consists a lot of biopeptide. Bad digestion also impact on the gut health due to the bad formulation in terms of nutrient or in terms of ingredient. Again, or bad cooking process, not right pH, not right enzyme, or bad gut microbiota. So you can provide those organic acid, probiotic, enzyme, or biopeptide, vitamin or mineral that can improve the digestion. And fermented protein also consists of organic acid and the biopeptide. In terms of gut disturbance, again, if you use a nutrient for the diet that less nutrient, low mycotoxin, or good freshness, high quality in terms of digestibility, in terms of the less contaminant, that can also help to reduce the gut disturbance. Or in terms of the protein, it should not be denatured or no spoiled feed, for example. So all these factors we need to consider in terms of improve the gut health. This is one of the frame I cut out from the study from Gu et al. 2019. How he suggests how we control the Vibrio acid and improve the stress resistance by the gut environment. So in the gut, we have the bad bacteria and good bacteria. To switch or to be favorable for the good bacteria, we need the natural product, the metabolite. For example, here I list out the shrimp health nutrient like carotenoid, nucleotide, beta-glucan, mineral, vitamin, prebiotic, probiotic, acidifier, short peptide, or some of the L-amino acid. These can help the gut to switch or to favorable the pathogen switch into the beneficial bacteria. This is one of the advice from the Google for from doctor from Google in human gut health, and you can see they suggest 7 things that you can do for your gut health or for kind of food for gut health. You see, it's similar. It's similar. Human being and animal, almost similar. So you lower your stress. You get enough sleep. Get enough sleep also something like to relieve the stress. Or you see, take prebiotic, probiotic, for example, or check for food tolerance. Especially you in terms of the food, they also mention the fermented food. Fermented food is not only for animal, but also for human being. So why we have to do the fermentation? Why the fermented food is good for human being and for animal? So first you can see the fermentation that like the bacteria pre-digest the protein and make it easier for stomach to digest, and it can minimize or completely eliminate the protein or make bioavailable for nutrient, or remove the anti-nutrient or harmful bacteria, or they can reduce or eliminate carbohydrates. So functional food in human also good because it's leading to deliver bioactive peptide that responsible for health benefit, and bioactive peptide in Asian protein feed fermented food have been reported for exhibiting can see antioxidant, antibacterial, or anti-diabetic, or anti-cancer. So fermented, that's why we do the fermentation. We need the fermentation product to help the gut health or help the stress resistance. And normally the proteins rich fermented food can result into enzyme, into different peptide, especially the biopeptide. And here you can see different function of biopeptide. It involves many health function. You can see antioxidant, immunomodulatory, or antimicrobial, and so on. So it's a very important because it consists a lot of biopeptide during the fermentation, result into organic acid, enzyme, and biopeptide. So, but we have different fermentation technology. You can do the fermentation protein by lactobacillus or by bacillus. By lactobacillus, it normally is anaerobic fermentation, but for bacillus, normally is aerobic fermentation. But lactobacillus anaerobic fermentation, there's a advantage that then can reduce the more anti-nutrient factor or cut down the protein amino acid into more short peptide to reduce the protein molecule, or produce more the organic acid and functional peptide. And organic acid is very important, that can inhibit the pathogen and make the mineral more bioavailable to be better utilization. Organic acid can also help the pepsin activity, or at the end, the protein digestibility improvement. Fermentation that can produce a lot of health nutrients for gut health and for health in general, like the short peptide, short chain fatty acids, reduction in anti-nutrition factor, increase the biomass, microbial cells, mediator, improve the digestibility, increase the protein concentration, or produce some special biopeptide that causes the bacteriocin. And these bacteriocin can eliminate the spore of the microsporidia. And also some of the antimicrobial peptide result during the fermentation. So here's some of the research. I show you the benefit of the fermentation that result into the organic acid and improves the apparent digestibility. For example, the fermented soy protein that can improve the digestibility in the shrimp and in the marine fish. Here's one of investigation from Science Chain Biotechnology. They showed us some of the bacteriocin produced from lactobacillus together with antimicrobial peptide and some other chemical can useful to kill the free spore of microsporidium. And today EFB is called by the microsporidium, and it's a very terrible disease for shrimp today, and very much concerned by the farmer today. So if you can use a fermented protein that consists of the specific biopeptide, that can help a lot in terms of reduce the spore of the microsporidium during the feeding. So that's why we do the fermentation of the corn protein, and we name that a MOTIV product. So that's why we call it a bioactive protein ingredient, because it's a fermentation and produce target to produce the bioactive peptide and organic acid also. So we also do the fermentation of MOTIV of corn protein concentrate by anaerobic fermentation with lactobacillus, and result into biopeptide, organic acid, and also the nature of corn protein concentrate also a lot of amino acid and mineral carotenoid also. Here they are the nutrient spec of the MOTIV, and you can see the protein somewhere 69%. We have 7.2% organic acid and very good amino acid profile. One of the limitations of the corn protein is low in lysine, but through fermentation, some of the lysine print up for this product. So lysine here, you can see is almost 3%, but the nature of the corn protein just somewhere 1% only. And we have very high carotenoid. It's around 285 ppm of Bigman xanthophyll, and also good metabolize energy. And it's a bioactive protein because again, it consists of the organic acid, and majority is a lactic acid. 7.2% of the product is lactic acid. And we have some nucleotide there. We have different peptide fraction. We have around 5.3% of the product is soluble protein or the short peptide below 10,000 Dalton or 10 kilodalton. Around 3.1% of the product is a bioactive peptide below the 1,000 Dalton. And also, again, we have a lot of carotenoid, and that can help to stress resistance or weight and survival. And here I summarize some benefit of that MOTIV product. You can see the B8 of product only 4.6, and this is very good to lower the B8 in the gut, to inhibit the Vibrio and also activate the enzyme to be better digestion. And also short peptide. We have no anti-nutrient, glycine, beta-conglycine, lectin, and oligosaccharide. All these anti-nutrient is not detectable in the MOTIV product. So it's a very clean product. And we don't have time to present you the trial, but we have many trial on stream, and just have one trial on the big leg. And through the trial, different trial, we have some conclusion on the product. Is first, MOTIV can improve the shrimp coloration when you fed the diet consists of minimum 7.5 in the formula. That can improve around 1.5 color score or around 12% color improvement. Through the different trial on stream, we see that it improves the survival related to the environmental stress and healthy gut. Around 4% survival rate increasing in the shrimp. And it's a very, very interesting that the trial we did on the piglet, it significant improves, reduce diarrhea rates and reduce mortality when we separate on the first phase of the piglet. When we separate from the mother, the mortality reduction up to 25%, and the diarrhea rate also reduce a lot. In general, it's improve the production performance in terms of lower FCR and the weight gain in the shrimp, around 5% in weight gain improvement and 5% lower FCR. In conclusion, I can have here, gut health play very huge role on the both mental and physical health to all animal and human. Again, managing diet and stress is a key strategy for gut health and for stress resistant improvement and overall health in general. Thank you. I finished my presentation here and looking for your question. Thank you.
Thank you, Hoa. It was a very clear presentation. We look forward in the next seminar to see some practical results of your product's inclusion as well. The next speaker is Martin Guerin, who is regional technical manager of Adisseo Malaysia. Martin has over 30 years of technical and commercial experience in aquaculture nutrition, and his work has been a lot in Asia, but also in Europe and the USA, formulating aquafeeds, supervising research and product development for both aquafeed companies and feed additive companies. He has worked also a period as a consultant, but joined Adisseo as regional technical manager for aquaculture 3 years ago, if I'm not wrong. and he supports Adisseo customers, feed mills and farms on technical issues and feed formulation, mostly in the Asia Pacific and South Asia region. The presentation title is Feed Additives Help Protect Fish Health, Fish Gut Health and Function, sorry. So Martin, please go ahead with your presentation.
Thank you, Hans. I'm not sure if you're seeing my screen at the moment. I think I put it up. You should be able to see it. Yes. Hello?
No, your screen,
No.
you need to share your screen. Yes, we can see it now.
Okay. Okay. Good evening, afternoon, or morning to wherever you are. I'm really happy to have been invited by aquafeeds.com, and I thank them to give me a chance to talk about how some feed additives can help protect the health and the function of the fish gut. Now, if you look at the health of the fish gut, I mean, you have to look at the health of the epithelium of the gut inside the intestine. And you have, in normal conditions, you have a healthy gut with a very diverse, well-established population of microbiota, which helps keep the pathogenic bacteria away and pathogens away. Then you have a protective layer of mucus, the epithelial cells, layer of epithelial cells that play a very important role in the digestion and absorption of nutrient. And below that, you will find in the lamina propria a number of cells that play a very important role in the immune response. The problem is, and I thank Dr. Hoa to have given us a very nice introduction on gut health. There's a lot of potential aggressors to this intestine. You have pathogens, the bacteria, parasites, but also feed components like allergen, anti-nutrient that can lead to a damage of the gut epithelium, leading to inflammation process and enteritis, opening of the tight junction between the epithelial cells, which can allow pathogens and toxins to penetrate through the epithelium and penetrate the organs below the epithelium. That's where you have an immune response with the immune cells, that you have inflammation as a result of this response. And this whole process leads to a loss of functionality of the intestine, which is a very important organ, not only for the digestion and absorption of nutrients, but for the immunity. Is one of the most important, one of the most important roles of the intestine is the immune response. So to this presentation, I'll talk about some additives, how they can help protect the function of the intestine, starting with a lysophospholipid-based product and then continuing with a phytobiotic. Now, we have a product, Aqualeso, which is a phytobiotic, which is produced by controlled hydrolysis of lecithin. So phospholipid have two fatty acids under the glycerol radical, and through phospholipase you can hydrolyze this, and that means you have only one fatty acid. So that molecule has less lipophilic component, so it's more hydrophilic, and that means that lysophospholipid can be very good emulsifier under to emulsify oil under aqueous environment, which is the environment that you find in the fish gut. So, as a result of this better emulsification properties, you have better lipid digestion and absorption. And that has led to very wide use of lysophospholipid to spare inclusion of lecithin and oil, as well as energy. So it's a known benefit, but actually lysophospholipid contribute much more than that. They can improve gut functionality, as I will explain, and improve the absorption of other nutrients, and overall stimulate the metabolism that is involved with the functioning of the gut. So if you look at the typical cell membrane, it's composed of a phospholipid bilayer with a water-loving side on both sides of the membrane, exposed to the aqueous environment. And in the middle of the bilayer, you have the fatty acids, which are hydrophobic. Now, when you introduce a lysophospholipid, and because lysophospholipid occur naturally in the intestine and are found in those cell membrane, you improve the flexibility and the permeability of the membrane. Now, when you look, lysophospholipid have only one fatty acid tail under the glycerol component. That means this leaves more room inside the membrane and more flexibility for small molecules to pass through for the passive diffusion through the membrane, as well as leaving more space within the cell membrane of the epithelial cell. So where you find a lot of the transporters, which are proteins, big molecules that can insert themselves inside the membrane. And therefore, if you have lysophospholipid, help accommodate those big proteins, which are responsible for the active transport of important nutrients like amino acids and other molecules. So through this presence of the lysophospholipid in the bilayer, you have you improve the fluidity and the permeability of the membrane, and therefore the passive and active transport of nutrients. And we have done some interesting experiments to demonstrate this benefits and functionality, working with a sea bream, which were fed for 4 weeks, low fish meal diet, high plant protein diet, therefore, rich in potential allergens and anti-nutrients that you find in plant proteins. So they were fed either the control diet with this type of formulation, or the control diet plus 1 kilo of Aqualeso per ton of feed. After this feeding for 4 weeks, we took samples of the intestine and established small section of the intestine, which we would place on wet mounts. We flattened so that we could put this wet mounts into special individual chambers where we can control the conditions of the environment of on both sides of the epithelial membrane. So that allows us to measure a number of parameters, both physiological and physical parameters. So one of them was to measure the absorption power through this membrane. So you can measure the flux of nutrients through the membrane that go by passive or active transport. And the intestine also secretes molecules like enzymes or the mucus. So if you want to boost the absorption, capabilities of the intestine, you want to make sure that the absorption capability, the flux of molecules through the intestine is increased. And we found that the fish fed the lysophospholipid had actually 3 times a stronger absorption capability compared to the control fish. We also look at the transport of amino acid by establishing the gradient of amino acid solution on the one side of the epithelial cells. It's supposed to be the external side of the epithelium found in the lumen, and look at how those amino acids were transported through the epithelium barrier into the other side. And by measuring the concentration of amino acid, we were able to determine that those fish which had been fed the lysophospholipid product had 2 times higher amino acid transport capability compared to the control fish. So what we were talking about earlier about lysophospholipid that incorporate into the membrane, they can improve the membrane fluidity and flexibility. And there, through this very nice experiment, we're able to demonstrate that they really actually boost the active transport capacity of the epithelial cells. We also run some, more general shotgun proteomics that allow us to look and study almost 5,000 proteins and looking at how they were upregulated. And that we look at the intestine and the liver and found that in the fish that had been fed the Aqualeso, they basically, you had a stimulation of the proteins involved with nutrient transport and the metabolism. So overall, very nice improvement on this metabolic patterns. Now, this is one type of additive, but we have long worked with another additive we call Saraco, which is a phytobiotic, which has been used for many years as a health additive to help fish farms to achieve better results because it positively modulates the gut health thanks to antimicrobial properties, which we have well demonstrated, and also improve the diversity of the microbiome in the intestine of fish like sea bream or sea bass, and we also demonstrated that with tilapia. We also run some studies showing that the additive can protect the health of marine fish infected by gut parasites like Enteromyxum leyi, which really damage the parasites. So we have improved survival. But in this experiment, our objective was to look more in details what is the benefit of using the additive on the function of the intestine and the epithelium of the intestine. So we look at whether you can, the additive could improve the integrity of the gut and also play an anti-inflammation role. So this was done through a feeding trial for 6 weeks where we fed a controlled diet with low fish meal again and high plant protein diets, again rich in allergens and anti-nutrients, and this controlled diet supplemented with the phytobiotic. At the end of the feeding trial, we took tissue samples and looked at electrophysiology of the intestine, as well as doing an assessment of the morphometric and histological parameters of the villi height and looking at what damage could have been done through histology on the intestine cells. So looking at the electrophysiology, what happens when you damage the intestines, you basically weaken the integrity of the epithelial cells. You widen the tight junction between the cells that allows passage of electrolytes, for example, and that basically leads to more easy, you have an electrical current. If you apply electrical voltage between the two sides of the epithelium, you can have electrical current passing through the epithelium. Now, if you have a more resistance, more healthy gut, you're going to have a higher electrical resistance, so allowing less passage of electrical current through these epithelial cells. So in order to do this, we went through the same methodology of using a wet mounts of a small section of the intestine as the one I explained earlier, placing the wet mounts in those individual chambers where we can control the condition on both sides of the epithelial membrane. And applying the electrical voltage on the side, on both sides of the tegument, we were able to demonstrate that the epithelium of the fish fed the phytobiotic had a significantly higher transepithelial electrical resistance in the interior intestines, up by 30%. So basically, you had a more resistance, more better condition of the gut epithelium. We also ran some histological evaluation through 2 parameters, looking at the ratio of internal perimeter, measuring the perimeter of the folds inside the gut, and the external perimeter, which is an estimate of the mucosal surface and therefore of the absorption capability of the gut. We also measured in the slide at the bottom, the ratio between the external diameter and the internal diameter. The internal diameter is the diameter of the empty space in the lumen of the gut where there are no folds of the no villi and therefore no absorptive capability. So by with a high ratio of external diameter over internal diameter, that means you have very small lumen, empty space in the middle of the intestine and therefore better absorptive capability. In both cases, through both those parameters, we were able to demonstrate that feeding the phytobiotics was boosting those ratio by a significant factor. It means improving the absorptive capability of the intestine epithelium. We also worked with probiotic like looking at we had established a gut histological score measuring various parameters and giving a score to those parameters, like measuring the amount of infiltrates in the mucosa, lamina propria, or submucosa, as well as measuring the thickness of lamina propria, which is a sign of inflammation. And basically accumulating all those scores, when you get a very high score, that means you have more damaged gut epithelium, and you have a low score, that means you have a good condition epithelium. In all sections of the intestine, we were able to demonstrate that feeding the phytobiotic to the fish helped reduce the score and therefore improve the health of the gut epithelium. And that was very well marked by looking at what happened in the lamina propria in terms of infiltrates and thickness. So by reducing the infiltrates and reducing the thickness, you demonstrate that you have less inflammation process of the lamina propria, where you have basically a reaction of the immune cells to try to defend the tissues. So very marked reduction. You know, you can see like the lamina propria thickness in the midgut reduced by 61%, and the level of infiltrates reduced by 21%. So very interesting result. We also ran some proteomics there to try to see if we could correlate what we know about the phytobiotic benefits on gut integrity, as well as on its ability to modulate innate immunity that we have shown in the past, like detoxification or reactive oxygen species, phagocytosis, or anti-inflammation proteins. So looking at all those proteins, we were able to demonstrate that, again, that the proteomics confirm what we knew, that the additive can improve the gut integrity mechanism as well as the immune response mechanisms. So that was very quickly some results that we showed that with two types of additives. I just want you to remember that, you know, the fish gut is always under aggression by a number of factors. They can come from the feed, whether the allergens, from the plant proteins, anti-nutrients, toxins, or from the pathogens due to the intensification of farming, which leads to a higher incidence of pathogenic aggressors in the environment. So in order to fight those, you can work with some feed additives that have documented function to support the gut integrity and functions, like lysophospholipid, that do more than just boost the digestion and absorption of lipids, because they also improve the absorptive capacity and transport of amino acids, and phytobiotics like Senacor, which have, yes, well-known antimicrobial properties and benefits to protect against gut parasites or pathogens, but they also improve the gut integrity and function. So even then, when you don't have a disease challenge, you still are benefiting from using those type of additives, because they improve the mucosa morphometry and they reduce the symptoms of inflammation coming from those aggressors. So that was a very quick presentation. I hope I didn't take too much time. I would like to thank you very much for your attention and look forward to your questions.
Thank you very much, Martin. You stayed exactly within the 19 minutes that we agreed before, so I'm very happy that you kept the schedule. And I think we are all, audience is happy to see how Adisseo helps to improve or to protect the gut integrity in the several animals, or at least the strategies that Adisseo has in mind. Our next presenter is finally, we stop with the old guys and we are going to listen to some more young professionals. The next speaker is Siméon Fagnon. He's Innovation and Product Manager of Phytsosynthese. Siméon is graduated in Aquaculture and Management of Aquaculture Resources at the University of Liège in Belgium. He spent 4 years in aquaculture research and project development in Benin, in West Africa, and joined Phytsosynthese 4 years ago, where he develops botanical products for aquaculture and provides technical support for aqua stakeholders in many countries around the world. Please, Siméon, take it away. You have to unmute.
You're muted.
Yep. Can you hear me?
Yeah, okay.
Yes, we can.
Thank you for this opportunity to give an overview of using Botanical in aquaculture, mainly to improve gut health. My presentation will be outlined as follows. Firstly, I will clarify a bit the concept of gut health, and then explain what are botanicals. After, I will explain how botanicals and show how botanicals can impact gut health when used as aquafeed additives. And finally, I will discuss about different challenge using botanicals in aquaculture. Gut health is a multidimensional concept which focus on different pillars, and I identify 3 main pillars here. The first one is good intestinal integrity, which is characterized by intercellular junctional complex. And the second one is effective immune status, characterized by different lymphoid tissue and other immune marker associated to gut. And thirdly is the gut microbiota, and then an established, well-balanced gut microbiota. All these concepts are interrelated because gut microbiota can influence intestinal integrity or immune status and vice versa. There are many factors that can affect or influence gut function. And as you can see, feed composition with the presence of different antinutrition factors. Like for instance, with when we fed salmon with soybean meal at certain level, it can induce enteritis. We have also stress, which we can enumerate a different kind of stress in aquaculture with this kind of intensive farming system we face. There are chemical, physical, and environmental factors that can impact also a gut system. Another factor is pathogen. We have various pathogens like bacteria, virus, parasite, which can also influence or affect the good function of our fish gut system. And finally, different toxins as well that can impact this system. Since some years, and mainly last year, I see that there are many interests in this topic, gut microbiota, gut immune system. And I think what is mostly important is that phytogenic as well is a review to evaluate the effect on gut health. It means that there is an interest on this topic and an interest on application of phytogenic in gut health improvement or gut health maintaining. what are botanicals for the opinion? Botanicals is a plant or a substance or compound which is derived from a specific environment, like a forest or wild environment. We can find plant in forest or wild environment. We can also find plant from cultivated area, like turmeric, for instance, in Asia. We can also find some metabolite from plant processed by bee, which are interesting in terms of botanical substance like propolis. And finally, we have algae in marine environment, not as a nutrient source, but as functional, for functional application, which can also categorize into botanical concepts. And we have a diversity of bioactive families. And as you can see here, we have plenty many plants. And inside of each plant, we have a huge diversity of molecules. And it's these molecules that constitute the exciting side of the botanicals properties. If we take, for instance, cinnamon, we can see cinnamaldehyde, cinnamic acid, many family of bioactive, which are very interesting to have potential functionality in fish or shrimp. Sorry. I don't up. Sorry. Now, to deal with this complexity, it's important to master what I call the phytochemistry, because it's important to know what are inside of this plant, what are inside of these botanicals. And to do that, it means that we have to characterize these plants. To characterize these plants qualitatively first, and then it means that we have to identify what are inside of this plant. And secondly, to quantify the content of these bioactive molecules in each plant. And it means that you have to maybe develop an analytical method or use at least a reliable method to make all these analyses. And what is important beyond that is how to formulate, how to combine different family of molecules to get a functional activity we want to get at the end. And it's what is quite interesting because it's different family of molecules and they have to be interact in order to get a synergetic property or activity. We can find about botanicals under many forms. We can find them on the crude plant powder or from extraction plant extract powder form or essential oils. And what is important is that there is a huge variation according to different factors. It's not only according to a different factor. It's not that we take one plant and there is the same content of active molecules. Here they can differ according to origin, according to organ, according to process, according to climate or season. And you will see here that from Sri Lanka to Brazil, we have different content of curcuminoid in turmeric. And the same when we use Boldo, you will see that there is different content of alkaloid according to which organ plant we use to characterize or to identify molecule inside. Now we will talk about how botanicals function, this variety of molecules can impact gut function. There are different ways, and each molecules or bioactive molecules family have their own properties. Some have the capacity to improve gut integrity. It means that they will reduce inflammation, they will improve tight junction protein, villi morphology. Others can modulate or balance microbiota. Others can improve immune system, and others can act on enhancing antioxidant capacity. And all these put together at local stage can improve functional gut. Here I want to show you how botanicals can impact inflammation, can reduce inflammation in gut. And I want to point out interleukin-1 beta, which is a pro-inflammatory cytokine, one of the most important pro-inflammatory cytokine. And it means that the increase of the long-term suppression of this pro-inflammatory cytokine can provoke a disruption of TEF junction protein and then increase the intestinal permeability. And it means that they will open, open the paracellular pathway for antigen, for pathogen, which will lead to disease outbreak. And here we have two groups of fish, post-salmon, fed with 25% soybean meal inducing enteritis. And you will see that after gene expression, 56 days of experimental, one group were fed with phyto product and other not. And you will see that we have a reduction of gene down regulation of interleukin-1 beta in the group fed with phyto. And as technical benefit, is the way to show that it's possible to reduce with botanicals compound inflammation, which will help to securize at least growth performance in fish. The second way is to show you how botanicals can also impact morphology in fish. And here is a publication on Nile tilapia. And we have two groups, the one fed with phyto, the second not fed with phyto. And the botanical compound were fed over 56 days. And histomorphological analysis, morphometry analysis show that there is increase of length of intestinal fold when fed with phyto. It means an increase of area of absorption, and at the end, it will improve the nutrient absorption with this botanicals compound. And at the end, we will have an efficient conversion rate of feed and at least maintain growth performance or even improve growth performance in fish or shrimp. It's also possible for botanicals to prevent for hyperpermeabilization, as I showed in the mode of action before. And here is an example on Pengzi Christian cap. And you will see that botanicals compound improve upregulated some gene related to TEF junction protein. I mean, Claudine, zoocludine and occludine-like, which are very important protein for the structuration of intercell cell. And here you will, as technical benefit, it means that there is a control of pathogen, prevention of pathogen to go through the paracellular pathway. And it means that they will less, the gut will be less affected and the growth performance will be maintained at least because overall health will be protected. The fourth example is how botanicals can impact microbiota. And on the same species, a microbiome profiling was performed at over 56 days. And you will see regarding the principal coordinate analysis that the group not fed with phyto product, phyto compound, show a wide distribution compared to the group fed with phyto, which is quite homogeneous. And regarding the microbiota composition, there is a difference between two groups, and abundance of Pseudomonas and Flavobacterium was low compared to the group not fed with phyto. It means that these bacteria are potential pathogen, and the low abundance will reduce at least the disease outbreak occurrence. And now at the end of this trial, there is a challenge with Aeromonas hydrophila, and there is in the group fed with phyto, we have low mortality, which means that using botanical compound, we can protect disease, we can prevent from disease outbreak, we can balance the microbiota, we can reduce opportunist pathogen in the gut of fish or shrimp. And finally, I will show you how botanicals compound can improve immune status and also antioxidant capacity. And this is an example on shrimp. And you will see how innate immune marker and antioxidant marker like phenol oxide, after 19 days of feeding with a phytocompound, was significantly increased compared to the control. Phenol oxide increase, which is a prophenol oxidase system in invertebrate. This increase, and you will see that lysozyme activity as well increased. And regarding malondialdehyde, which is a lipoperoxidation metabolite, this marker decrease. And it's the way to show that botanicals can have an activity not only on fish but in shrimp, but they can also have activity to help to improve gut function by improving the defense capacity, by improving antioxidant capacity, by promoting the secularization of production system, because we will have low mortality in case of disease outbreak, and we will have a higher survival rate. Now, I want to mention some challenge of using the botanical compound in aquaculture, because in aquaculture we use extruded feed. Extrusion is a key process in aquafeed manufacturing, and here I list some essential oils and show you the flash points. It's you will see that these flash points are under 100 degrees. It means that if you use these essential oils, it's possible to get it's possible for them to evaporate the bioactive molecules, and then you will see that there won't be any molecules inside like here. Then what we have to do, the importance is to protect these molecules from evaporation, and the good way to do is to encapsulate them. And here is an example of eucalyptus, and you will see that over 12 minutes when challenged with 130 degrees, there is no cineol in the eucalyptus in the product. And that is important to protect when we are in phase of, we use essential oils. Essential oils are more volatile compared to over technology, crude powder or extract. But we need to consider that in the way that we work with botanicals. And I want to also talk about dosage and which dosage and the synergy are relevant in this kind of activity. What is important is not to increase dosage. It's not to add more botanicals, but what is important is to identify the right dosage according to species, according to system, according to many factors. And what is mainly tricky is how to find the synergy interaction, how to combine different molecule family in order to get a functional activity at the end in the gut and in the overall health. Do botanics cost effective in aquaculture? Yes, if we reduce, because increasing dosage does mean that you get more activity done. Don't waste a product, or don't add more product for nothing. And different technology exists to manage all this. As I said, essential oils, plant powder or crude plant powder, all these are interesting to be explored. And now I want to talk about regulatory issue and environmental issue in using botanicals. Just to say that botanicals are well authorized as a feed additive or premixture or complementary feed. They are authorized by EFSA. And in terms of environmental safety, attention should be paid for marine environment. And also we have using botanicals mean that they can be a risk. We can find some risk which is related to undesirable substance. And if you read this publication, you will see that we can find, for instance, pesticide risk in this kind of activity, and we need to implement a good management strategy to tackle all these risks. As take-home message, I want to say that fish and shrimp homeostasis depend intimately to the gut health. The gut health is very critical, important, and I show many parameters: morphology, permeability, microbiota. All these parameters are very interesting, and the knowledge, the phytochemistry knowledge, is important to understand what's inside of botanical and how we can make interactions of these molecules to get activity, functional activity. They are rich, botanicals are very rich, very diverse, and the phytochemistry is important to deal with that. Finally, important is the regulation, the regulatory issue, which is a key for sustainable application of botanicals in aquaculture. Thank you for your attention, and I'm open for any question.
Thank you, Simeon. Also, you managed to stay within the 19 minutes. It's really comforting to see that also phytobiotics can help our animals to deal with all the stressors in the gut. Then we will go over to our last presenter, Mr. Valentin Eckhardt, who is a product manager of Aqua Applications at Biochem. Valentin graduated with a master's degree in aquaculture science from the University of Rostock in 2016, after which he led the EU project InnoAquatech to develop innovative and sustainable aquaculture technologies for the Southern Baltic region. And today, Valentin works as R&D application manager for aquaculture in Biochem in Germany, in Lohne. And his focus is on the application of feed additives in aquafeeds to improve the health and performance of fish and shrimp. Please, Valentin, go ahead.
And thank you, Hans, for the introduction. And also thank you very much to the Aquafeed magazine and Lucia to giving this opportunity to speak here, and also welcome to everyone over the world. Yeah, thank you. I will now start with my presentation, low dosage, big impact, yeast effects on gut health. Well, if you have not listened carefully to all the presentations ahead, well, why are we talking about gut health? Because, well, I think it's very obvious that the fish gut is the main entrance for transmission for pathogens. So we need to understand this route of transmission and how we can affect this portal of entry. And well, there are some intestinal barriers against pathogen transmission which can be affected, and we have heard already about some of these, like the intestinal mucus layer is one very important. Here we can, for example, increase mucus secretion via, for example, increasing the density of mucus glands. On the other hand, we can also have an impact on the microbiome. So here we have also a big barrier. We have heard already about this. So we want to sustain a stable and balanced and beneficial microbiota to really protect the intestine of the fish and shrimp. But today I want to focus more on this biome part. One thing which can, for example, destroy such a biome, which is called a dysbiosis, can be, for example, the use of antibiotics. So I think we all agree on that this will completely clear parts of the intestine and therefore make room for colonization of some pathogens, and therefore should not be a solution for the future. But of course, also environmental changes can have an impact here, but also dietary shifts. So I will give two examples. So for example, for environmental changes, there was a very interesting publication recently given in the online presentation during I think it was the Aquaculture Europe 2020 event, where they showed some results on Atlantic salmon during the freshwater phase and the seawater phase. And what they have found here, it's very interesting that we have a very hard shift in some bacterial species, like for example, Lactobacillus was found in the freshwater fish, but not in the seawater fish. And on the other hand, like Vibrio bacteria were less found in the freshwater phase, but during the seawater phase, these have increased. So here the environment definitely has a very strong effect on the gut of the Atlantic salmon. And then there's a publication from Silvia Torrecilas. I think I have seen that she's also participating today, so I hope it's okay that I present your data here, because I found this is really interesting. So I just picked some data from the study, and what was interesting, for example, here they were comparing different dosages or inclusion levels of fish meal and fish oil fed to European sea bass. And then where they were looking also on the microbiome, and what I picked here, I just picked some species, but there are also other bacterial species shown, and all this together shows a very big shift during the change of fish meal and fish oil in diet. So here you see the fish meal and the fish oil content. So this was the control group they used here, and then continuously reducing both. And as you can see, there were some bacteria, like for example, Clostridium or Dermacoccus, Lactobacillus and Staphylococcus, which were increased due to this shift. But another thing which was not very much highlighted in this publication, but what I think is very interesting, is that there are also the non-identified bacteria listed, and this relative amount was highest for those with a fish meal diets in there. And I think that's maybe a sign for a more balanced microbiome, because you have found maybe less of those identified bacteria, because they maybe were under the threshold for analyzing. So when you have more pronounced bacteria here, this means that these are more prominent, and this is more a sign of maybe a imbalanced microbiota. On the other hand, these effects were also seen, or that there were also a shift in the morphology of the gut. So here, for example, they also analyzed the goblet cells in the fold area from the posterior intestine. And also here you can see an increase of the goblet cells for more produced, or for an increased production of mucus, which could be, for example, a protection to a shifting microbiome, or maybe also an interaction with more lactic acid producing bacteria, which then also triggered an increased lactic acid production in this area. Also, the submucosa width was increased, so there were definitely some linkages between the morphology and the shift of the microbiome inside of the gut due to the shift in the diet. I think the most prominent, and which we have heard today already sometimes, are the enteritis results that have been seen so far. So here, for example, some pictures showing the corn gluten meal induced enteritis in turbot with different diets, with the fish meal based diets, and then different levels of corn gluten meal. And you can relatively easily see that the microvilli length in the fish meal diet was much higher, also microvilli density, and well, for the other ones it was reduced, well, which means a reduced surface area, reduced area for nutrient uptake. So definitely a sign of enteritis. And I think the most prominent we have seen so far is the soybean meal induced enteritis here in Atlantic salmon. So you can assume with such such a gut epithelium, it's hard to take up the nutrients. So I think it's a very good picture showing that you want to have a healthy gut, definitely. So coming back to those intestinal barriers against pathogen transmission. And here, what can we do? For example, for the mucus layer is, for example, beta-glucans in the past have shown to have an effect on a higher density of mucus glands and higher mucus production inside of the intestine, but also organic zinc supply, for example. But these have a direct effect on the host. So, well, it is effective, but you should be careful in dosing, because especially here, when you have an overdosing effect, maybe this can cost performance, because the animal needs to produce something new inside of the gut, like producing more mucus glands and so on. The other thing is the use of prebiotics and probiotics, which have a direct effect on the microbiome. So here, interacting and modulating the microbiome is more an indirect way, and probably a little bit less challenging for the animal. So you would not assume that there is, for example, an overstimulation or something like that. Today I want to talk about this later part, so more on the yeast side, and as yeasts are prebiotics or sometimes also used as probiotics. Here are some differentiations. Live yeasts, commonly known as probiotics, but then there can be separated parts of it, like for example yeast cell walls, like our product Technomos, for example, which is an autolyzed yeast cell wall product. The inner part can be also used, which are then yeast extracts. There are also hydrolyzed yeasts, like our product, for example, Techno yeast. But on the other hand, there are also completely killed and inactivated yeast products, like, for example, dead yeast from brewer's yeast or yeast cultures. There you have the inactivated, for example, heat and inactivated yeast cells, but also sometimes also parts of the cultivating medium inside. Let's take a closer look into the yeast. Well, you have all the common cell organelles inside of the yeast. The functional ingredients are, for example, found in the RNA as the nucleic acids, which can be broken down to nucleotides, but also in the cell wall, where you have the plasma membrane, you have the inner cell wall, the outer cell wall. So in the inner cell wall, you have the beta-1,3,1,6 glucans, and in the outer, you have the mannans and also some proteins. So let's talk about these mode of actions of these functional parts of the yeast. I tried to draw a picture here on the left-hand side. You can see the gut epithelium, you have the gut lumen, you have the lamina propria, and this should be some kind of an infestated microvilli. And well, you have the gram-negative bacteria, gram-positive bacteria, some viruses, and they try to break through the epithelium by degenerating the epithelial cells, widening the spots between the tight junctions and so on. And here the main thing I want to explain is also those type 4 fimbriae, especially for the pathogens like Vibrio, for example, gram-negative bacteria. In other cases, also type 1 fimbriae can be used to attach to the intestinal surface, and as soon as they attach to it, they start to proliferate and start to colonize the intestine. So if we look on the other side, so when we use, for example, beta-glucans, there we assume that we have a stronger gut epithelium already because we have a protection coming from the beta-glucans. So also here we have still an intact mucus layer. And this is also influenced by a microbiota, which is still there existing, not inhibited by pathogens. But let's come to the beta-1,3 and 1,6 glucans. What are they doing? They can be recognized from, for example, pattern recognition receptors and so on from dendritic cells as some kind of like a pathogen, but even when they are not having any pathogenic effect, they are triggering the immune system, starting to send out cytokines. For example, starting the differentiation of monocytes to macrophages, which then in turn start to engulf and digest pathogens like the viruses. And then when they have found something, they can also use the beta-glucans as something that they find, and then they can emit also cytokines to attract more macrophages to have a very strong immune system effect just on spot to get rid of the pathogens. When we look into the MOS, the mannan oligosaccharides, we have, well, the same situation, but the effect is different. So we have the MOS particles, which are commonly indigestible by the host, but which can be used as substrate by the microbiome. So here bacteria can use it, also digest it, and by there you're definitely helping also the microbiome to grow. On the other hand, you have also an effect on the pathogens, especially the gram-negative bacteria, for example, where the MOS particles can bind to the binding sites, block these from binding to or adhere to the intestinal wall, and excrete these later on. So hereby you're lowering the pathogenic pressure, and that's very important to sustain a stable and good gut health. These are, for example, some pictures showing how these MOS can affect, well, a good development in the end in the gut epithelium. So here you see, for example, with MOS and without MOS, the length of the microvilli, but also the microvilli density. So there are definitely some good effects coming from MOS content. The last functional ingredient I wanted to mention is the nucleic acids or nucleotides, which are very relevant for recovery. After, for example, an infestation, because nucleotides are necessary for building new DNA during cell division. So every part where healing process is taking place or growth is taking place, you need a high amount of nucleotides, but also for the transfer of information from the nucleus, for example, to build new proteins. These nucleotides can then come from, for example, the RNA out of yeast product. When you have a whole yeast product, for example, a hydrolyzed yeast product, are broken down by the RNAs, and then they are helping to develop, redevelop, or healing the gut epithelium. But of course, also here, there's a sweet spot. For example, these are some data shown for rainbow trout, where different dosages of nucleotides were used. And you can see, for example, that, well, in the range of 0.05% to 0.15% nucleotide supply, you get a quite good experience, so good specific growth rate for the fish, but also reduced FCR. But then over the top, if you're still continue dosing, you can end up at a depression. So there the specific growth rate is going down again, and also the feed efficiency is going up. So depending on the product, in our case, well, when you use our product, about 1 to 3%, it's a feed material, not mainly a feed additive. You will end up at this specific dosage you commonly want to have. But coming back to the moss part, so here the yeast cell wall product, especially for having a very effective product, it's important that this is very homogeneous and very fine in its particle size. So here you can see, for example, a byproduct yeast, where you can see higher particle sizes compared to a primary grown yeast, where you have a very fine quality, uniform quality. And why is this important? So here, this is the idea behind. So when you have a primary grown yeast, you commonly produce this yeast just for the purpose of being a prebiotic. When you have an ethanol yeast, a byproduct yeast, these yeasts are growing to produce ethanol in the first place. And during this time, these yeast cells need to protect itself against the ethanol environment. So therefore, for protection, they are building up a thicker cell wall. And of course, later on in the processing part, hydrolysis and autolysis is not very effective when you have such a thick cell wall. So some of the parts still can stick together, are covered maybe in the surface area where binding sites might be for binding pathogens. So in the end, you have a lower pathogen binding effect. So, but is this also seen for real? So let's see. We have performed, for example, here a sedimentation assay. So what we have done is we have had suspension with bacterium. So we were doing this with Vibrio campbelli, which is the pathogen commonly known for inducing luminous bacterial disease in shrimp. And so we have had a negative control where we have had just the bacterial suspension. Then we have the suspension with the yeast product from a byproduct yeast and with our product, which is a primary grown yeast. And then we were also adding the bacteria to it. And you can see that there was a higher sedimentation in that case. And well, it was about 80% higher compared to the byproduct yeast. So here, definitely more binding sites available to bind those pathogens. This was also then analyzed under the microscope, and here you can see on the left-hand side, again, the byproduct yeast, and even what I was trying to show with those drawings, you can see also here that there are those coverings from, it can be some remaining parts of the media, of the medium, which is covering the binding sites. So this was then unable to bind the pathogens. Here you can see the Vibrio parts in here to bind to the surface of the yeast cell walls. On the other hand, you can see with a primary grown yeast, well, you need to think of this as a 3D sphere, which is then pressed together for a 2D layer to microscope it. So there was a very nice sphere of adherent Vibrio around it, so very effective. And this layer around it was also measured, and here we can see a very constant thickness of this layer for the primary grown yeast, while this was also quite constant through all the samples. And here you see a higher standard deviation compared to a byproduct. So also here, the yeast quality is very important, what you select to have an effective product. So what you can do with a yeast product, with a yeast cell wall product, is that you can reduce the pathogenic pressure by, when you have a very effective moss content in it, by removing those pathogens, which improves the gut health and increases immunity. And well, I decided to use the title low dosage, big impact. So in our case, it's only 1 kilogram per ton, which has already a great impact on the fish side or on the shrimp side. In summary, well, there are several parts or several aspects which can influence the gut microbiome. So, for example, the dietary formulation, the environment, and also the use of antibiotics. Yeast products derived from yeast have a lot of functional ingredients which positively affect gut health, like mononucleosaccharides, beta-1,3,1,6 glucans, and nucleic acids or nucleotides. And in the end, there is a big difference in which product you're choosing to get a very pronounced effect. So thank you very much for your attention. I want to also use this moment to give a little, or to make a little advertisement for our upcoming webinar in the end of this year. I think this will be also advertised through the Aquafeed magazine in time. So we will have a guest speaker, Margaret Overland from the NMBU, talking about the secrets of yeast in aquafeed. So you're welcome to join this as well. And if there are any questions in between about our aqua concepts, you're free to ask me. Thank you very much. And I think I hand over to Hans again.
Thank you, Valentin. Also, you managed to stay within your time, but altogether we managed to almost consume all the time that was meant for this webinar. I think it was 4 very interesting presentations, and also looking at the response from the audience, I can see that there is a lot of questions raised by these presentations. I can assure each of the participants that put a question will get an answer from the presenters, but I think we can steal a little bit time to do some questions at least. And the first one that I would like to raise to all the presenters, actually it was Luis Alejandro Daki from Cargill, Ecuador, who put this question forward as well, and actually took the words right out of my mouth. What is your recommendation to apply these very nice concepts, either through feed or at the farm? Can you each respond to that? Maybe we start in the same order of the presentations. Dr. Hoa?
Yes, thank you for very nice question, very good question from Luis, my colleague from Ecuador. First of all, is for the MOTIV proteins, normally we apply in the feed mill, not at the farm. But for some other additive, like for example, the enzyme or probiotic or the other additive, you can top resin at the farm, especially in the liquid form. Anyways, some of the additives are better to add it in the feed mill, but some additive we cannot add in the feed mill, then we can top resin at the farm, because heat resistance is one of the limitations to apply in the feed mill. For example, enzyme, you have to develop the equipment that can spray the enzyme at the coater after the cooling. Or some of the probiotics, they cannot accept the heat, very high heat during the cooking. So especially the lactobacillus, you have to apply for the top resin at farm. But for other like sperm forming bacillus, you may apply in the feed mill. Okay. So different product, different application. Yeah.
Thank you, Hoa. How about you, Martin? What's your recommendation?
Yeah, I think Dr. Hoa answered in a nice way that there's a different additives deserve to be handled different ways. What we found is that also sometimes with the feed additives that we arrive, we can combine applications in the feed mill applications. We work with feed mills with specific type of additives that support the health of the fish or the shrimp. But when you have a specific challenge coming up, sometimes it's very difficult to react by the feed mill to a specific challenge. And so we have programs where we combine of a preventive strategy through the feed, either at the feed mill or the farm. And then we have a corrective approach that is handled by the farmer through specific. So for example, in shrimp, we have a very good example with Senacor where we work very closely combining this preventive and protective approach.
Okay, thank you very much. Siméon, what's your view on this?
Yes, I think both strategy can be applied. It's possible to use botanicals in feed mill and in on farm, but it will. We have to adapt. It's important to adapt the requirement for the need of a farmer or feed miller to the technology. We cannot use essential oils in liquid form, and then it's very suitable to be used on farm. But if you want to use essential oils on feed mills, I think we will get very quickly the limitation of heat stability, and we will need to encapsulate at least, or to have equipment to be used after extrusion, frost spraying. And it's different. It depends on what technology you have, how you formulate your product, and is it in powder, is it in liquid form, all this. But it's possible to apply botanicals in farm and also in feed mill. Thank you.
Okay, thank you. How about you, Valentin?
Yes, well, I think, yeah, I agree with my colleagues before. So the thing is, it depends a little bit on the species, and it depends a little bit on the pellet. So if the pellet has a very firm surface and it's hard to add anything on the surface, and you're working with a powder, I think it's a little bit challenging. But if it's a little bit more sticky, the surface, I think we have done some field trials where you can also add this with a mixer and add the powder, and it's then on the surface. But then in the next step, it depends on the species. So if you have a species which is eating the pellet right away from the surface, then it will end up in the animal. Otherwise, when the animal needs too long to feed on the pellets, well, then they will maybe dissolve in water. Like, for example, in case of shrimp, maybe it's not the best way. Of course, you will get always constant and good results when you add it to the feed directly.
Yeah, I think I tend to agree with that. And although you see that a lot of shrimp farmers indeed are doing a lot of coating, and maybe that is a message to the feed producers, because they are doing that themselves, knowing that maybe it's not so efficient, but basically mistrusting the feed supplier that they are putting the additive that they are requesting. Okay, that's my interpretation, but that's also what I hear in the field sometimes. Okay, maybe I stay with you, Valentin. What you're mentioning this low dosage really and the big impact. Is there any limitation on, let's say, on maximizing the amount of MOS? Can there be a negative impact of, or let's say, overstimulating the immune system, or how do you look at this?
Well, this depends a little bit on which MOS product you're using. So because when you just focus on a product which has some purified MOS, which is not something I haven't seen so far, but so just focusing on the MOS as a functional component, I have not seen any overdosing effect so far. But the problem is that most products are yeast cell wall products, and this means you have the whole yeast cell wall in there. That means the other functional component, like the beta-glucan, is also there. And as I said, the beta-glucan is used with a direct effect on the host. So the host is boosting its immune functions naturally due to reacting to the beta-glucan. And there I have seen already maybe some overdosing effect. So a basic dosage is really good to keep the level of immunity high. But if it comes together with a MOS, you always need to have a view on the beta-glucan content as well. So having both together is a very good interaction. So it would be good to look for a product which has a high MOS content and a medium beta-glucan content to have a good supply of MOS. And also here, well, if you have, for example, a product which is, as I have shown in my presentation, less hydrolyzed or autolyzed, so more sites are covered, then probably you need more of the product to have this effect. But it's a product related question, so I think that answers it a bit.
Absolutely, that answers the question. I also have another question for Siméon. You have really enlightened us with all this phytobiotics and these phytomolecules that have their different impacts. How do you see the combination or the potential combination with other gut health promoting additives? So we've heard about the MOS, for example. We've heard about other organic acids. Do you have any experience with that? Can you share something with that about that?
Yes, it's possible to combine botanical compound with other kind of additives, organic acid or probiotic or prebiotic. But I think what is important, first of all, they have all almost different mode of action. And then if we can find a synergetic interaction between them to get a functional activity, I think is quite good. But what is important is to consider the dosage, because it's not the right dosage and the right interaction between all these additives. But what is also important to mention is to avoid to combine two different botanical compound products, because we can lead to overdosage, and maybe we can get a contrary effect. Instead of get a beneficial effect, we can decrease or get pro-oxidative activity. And what is important, yes, it's possible, consider dosage, consider synergetic effect, and also avoid to combine different products, because it's a kind of biomolecule, different family combination.
Right.
Thank you.
If I understand you correctly, it can also be, let's say, tricky to combine, yes, molecules with different or similar actions.
We already have many molecules in one plot.
Yeah,
and then,
if you add something extra, it can be too much. So you have to test it
yes,
properly.
Yes.
Okay, excellent. Let's go then to Martin. One of the striking things in your presentation was basically you were presenting two products which were quite different products, each of them. Do you recommend them normally to use them separately, or do you also recommend them to combine them? Is that okay? That is also one of the questions from the audience.
Yeah, thanks for the question, Hans. Yeah, I mean, actually, both products can be used jointly without adding too much cost. If you look at Aqualeso, it's lysophospholipid. So when we use it, sometimes it's used to reduce cost in the formulation. So when you use it, you're not adding cost to the formulation because you're sparing energy. You're helping to reduce oil content. So in the end, the benefit I was showing are side benefit in addition to the main use of the product.
Okay.
It can be combined with the other, which is really a health products. Typically, farmers which are facing disease challenges are using the product and focusing on. So, no, each product has a different function. They can be used jointly, and yeah, there's no counterindication of using both. And since also you could see from the protein activation that we did on both, they work on different metabolism pathways. So they can be complementary. So.
Okay, thank you very much. And then I think a last question, unless some very burning things come up, to Dr. Hoa. You make your presentation about the use of fermented corn products. And as you know, in the market, we also find, let's say, competing products. I think they are competing products, fermented soya, hydrolyzed soya, hydrolyzed whatever, I mean, hydrolyzed is also one of the magic words these days. Could you elaborate a little bit on the maybe the similarities or differences between your products and the ones that I just mentioned?
Yes, thank you, Hans. Very good question. Again, corn protein concentrate and soy protein concentrate is in general, they are very good for the combination to balance in terms of amino acid. But in terms of fermentations, product like MOTIV, like you see in my presentations, we have no anti-nutrient and very clean product, no anti-nutrient. And especially the fiber and the carbohydrate, we are very low. The starch also, we are very low. It's almost pure protein. The starch, we on maximum the 1% starch and 1.7% carbohydrates. But if you look back on the own soy protein, including the hydrolyzed soy concentrate or the fermented soy, you will see it's not totally pure protein. It's still very, very high carbohydrates. Somewhere 23-24% carbohydrates still there, and lower protein compared to the MOTIV. And they still have the anti-nutrient there. Of course, different technology can bring back the different benefit among soy together also. For example, if you have fermented soy that fermentation by the anaerobic lactobacillus, there's some benefit there because they have the organic acid there. But if you get the fermented soy that fermentation by the aerobic bacillus, and then you don't have organic there. But you have another benefit for that fermentation is you may have the spore famine of bacillus there. To get the benefit of probiotic. However, aerobic fermentation could have the inconsistency quality compared to the anaerobic fermentation of lactobacillus. So different, even fermented soy, they have different quality, different product. So again, compare with MOTIV, we have more pure protein and no anti-nutrient. We have higher the biopeptide there. Yeah.
Sorry to interrupt, but is that because the MOTIV is produced of corn protein concentrate?
Yeah,
it's already concentrated before fermented. Is that correct?
We have very unique fermentation technology from
sorry, you cannot tell us.
From two different protein stream. Yes, it starts from yeah.
Okay, thank you very much. Okay, I just still one more minute, a last question to Siméon. One of the participants is asking if there is a specific method to measure phytobiotics in feed. Is it possible at all to measure in feed? I don't know whether it's useful at all to measure in feed.
Interesting question, but I think from my experience, it's very complicated to measure phytobiotic compound or botanicals compound in feed because it's quite complex matrix feed. Then there is no possibility to, there is no reliable method to measure the compounds in the complex matrix of feed. Because in feed we have different ingredients, and then with the low dosage of compound we incorporate in feed is very tricky to measure this compound. But
okay, I think that's a clear answer. I mean, no possibility is also an answer, right? I mean, it's not.
Yes, possibility is quite limited.
Okay. Well, thank you all thank you, Siméon, for this answer. Thank you all presenters. Thank you, audience. I think we had a very interesting almost 2 hours altogether. And we still have 140 participants at this moment. So it means it has been maybe a little bit long, but still worthwhile for people to stay on. And of course, it will be viewable later on YouTube. I give the floor back to Lucia. Thank you very much for organizing this nice seminar.
Thank you, Hans. Thanks for the great job moderating, and thanks all the speakers for the great presentations. I think the audience love all your presentations, and thanks MOTIV for sponsoring the webinar. And those questions that were not answered live, they will be answered by email. And thanks everyone for joining in. Don't forget to subscribe to our publications on our website, aquafeeds.com, and follow us on social media. All the webinars will be available, as Hans said, on YouTube for long-term use. And see you next time. Goodbye.
Thank you.
Thank you. Bye-bye.
Thank you very
Bye-bye.
much.
Bye-bye. Cheers.